PDGF cell signaling pathway by Arthi Chandra and Katherine Yang. So the first thing I'm going to talk about is identification. PDFG is a hydrophilic signaling molecule, meaning it cannot cross the cell membrane. Its receptor proteins, also known as the PDGF receptors, are located on the cell surface, specifically as transmembrane receptor tyrosine kinases, which initiate the intracellular signaling cascade upon ligand binding. Its mutation occurs in the same location and is also known as TEL-ETV6 activation. Purpose of PDGF, also known as platelet-derived growth factor. Like in the name, it regulates cell growth, division, and survival, such as fibroblasts, smooth muscle cells, and certain glial cells. It plays a key role in processes of wound healing, tissue repair, and angiogenesis. Dysregulation or mutations of this pathway are linked to pathological conditions such as cancer, fibrosis, and other dangerous diseases. Now to the advantages and disadvantages. Let's start with the advantages first. If everything goes smoothly, the first advantage would be that the pathway promotes tissue repair and regeneration by stimulating cell proliferation and migration, particularly in wound healing. The second advantage would be that it supports angiogenesis, aiding in the formation of new blood vessels necessary for tissue growth and repair. The third advantage is that PDGF helps maintain homeostasis in connective tissue and vascular systems by regulating the behavior of fibroblasts and smooth muscle cells. These three functions are crucial for maintaining normal cellular function and tissue integrity. Now, back to the disadvantages. Unfortunately, this cell signaling pathway has a 5 to 10% chance of mutating, which leads to our first disadvantage, being that it can increase the risk of cancer due to abnormal cell proliferation and survival leading to tumor growth. The second disadvantage would be that if the pathway has excessive activation, then fibrosis will develop, which results in abnormal tissue scarring and organ dysfunction. The third and final disadvantage would be that when PDGF promotes smooth muscle cell migration and proliferation, atherosclerosis progresses, which contributes to the plaque forming in blood vessels. These negative outcomes arise when the pathway is overactive or improperly regulated. Now the question is, how does this group of receptor proteins work? Well, under the circumstances where there's no mutation, the PDGF signaling pathway should work through 6 steps: ligand bonding, dimerization, autophosphorylation, signal transduction, cellular response, and termination. These 6 steps will further be explained later.
So what exactly happens in the PDGF, or platelet-derived growth factor, cell signaling pathway? I'll be going over how it's supposed to work through ligand binding and normal activation, and how sometimes it can go wrong with constitutive activation, although normal activation is more common and really should be occurring. But first and foremost, I will identify everything present in the pathway so there's no confusion throughout the video. Number 1, we have our receptors. The normal receptor protein is called the platelet-derived growth factor receptor and is a type of receptor tyrosine kinase. The mutated receptor is also a type of receptor tyrosine kinase. However, it is one important factor that makes it so different from the normal one. This important factor is TEL-ETV6, a proto-oncogene regulating different cells' growth, development, and differentiation. Number 2, we have the ligand. The ligand is used for the normal receptor, but is not used for the mutated receptor. This ligand causes the normal receptors to change shape to cause activation. The mutation is self-activating, meaning it does not use a ligand. Number 3, we have our mechanism, or mutation mechanism. The ETV6 or TEL gene translocates with the PDGFR gene. This process then gives us the fused gene called oncogene, which can lead to a lot of genetic harm. Number 4, phosphorus is another key part of activation in both normal and constitutive. When phosphorus crosses tyrosine kinase domains, it finalizes the activation of the receptor. There are 2 ways activation can occur. First, through normal activation, which results in an overall positive response for cell growth. Then we have constitutive activation, which overall results in a negative response that can cause a lot of harm and genetic diseases. In normal activation, we have a plasma membrane, on which two monomers of the PDGF receptor are present. Both monomers have inactive tyrosine kinase domains in the cytoplasm. Then, the PDGF molecule or ligand binds both the monomers of the receptor together. This leads the activation of the receptor and causes it to change in shape. After this occurs, cross-phosphorylation of the kinase on the receptor happens to activate the receptor further. This is the unmutated process that does not create the oncogene and instead aids in cell growth and healing within the cell. In constitutive activation, we also begin with the plasma membrane. However, now the receptor is embedded with TEL-ETV6 and is formed through transcription after the translocation between the TEL-ETV6 gene with the PDGFR gene. In this mutated process, the receptor can activate itself and change its shape without the use of a ligand. This is also known as constitutive activation of RTK and can phosphorylize by itself as well, and is naturally self-activating. This can lead to the creation of an oncogene, and an oncogene can lead to cancer and other extremely dangerous diseases for the body. Next, we'll be going over termination in the two different types of activation pathways. In normal activation, termination occurs through mechanisms that stop the signal to ensure proper cell function and prevent overactivation. This process unbinds the ligand due to its weak bond, which helps the cell's growth. Opposingly, in the mutated pathway, termination leads to uncontrolled cell growth. Unlike what's shown in the video, the monomer of the receptor proteins stay together and lead to overactivation. The PDGF signaling pathway primarily promotes cell growth, survival, and proliferation. It is involved in wound healing development and the regulation of blood vessel formation. However, any mutations like the example shown above can lead to diseases like cancer. Thank you for listening. Hope you learned something new.